Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Okay, well, good afternoon, and welcome to the MIT Faculty Forum. I'm Evan Morris, and I'll serve as the moderator today. For background, I'm a biomedical engineer by training, and these days I'm a professor of radiology and biomedical engineering at Yale. As you'll hear a little bit later, I study the brains of people addicted to drugs using brain imaging technologies. And what's nice about this forum that we have today is that my fellow panelists and I span the spectrum of research from basic to applied translational with humans to clinical applications. As a reminder, we welcome your questions during this chat. For MIT alumni joining via Zoom, please use the Q&A feature found on your toolbar. For all others listening on YouTube, you may add your questions to the comments field. We also encourage you to tweet using hashtag #MITBetterWorld. We'll get to as many questions as we can. Today we hear from our three experts, well, two experts and me, and we're going to start with Dr. Nune Martiros, who's currently a postdoctoral fellow at Harvard University.
Hello, everyone. I hope you can see me. I'm a postdoctoral researcher at Harvard here in the Murthy Lab. And before that, I was a graduate student in Anne Graviel's lab at MIT, studying basal ganglia circuits and how they contribute to habitual behaviors. So I'm just going to tell you a little bit today about the work that I did in Anne's lab. And let's see if I can get some slides. Okay, so I'm going to do share screen. And I'm going to go to view. So let me know if this is a problem if you can't see it. But what I wanted to talk about is that whether you may realize it or not, most of our life is made up of habitual action sequences. And most of the time, this is a very good thing. So things like making your coffee in the morning or tying your shoelaces, or for example, riding your bike from your apartment to your lab every day. We do these actions in a very stereotyped manner. And this way we can kind of allow our attention to go to more novel tasks that are more difficult. For example, writing our dissertation, which is not habitual at all, and it requires a lot of mental resources. So what actually happens when behaviors go from you first learning them to them becoming habitual? So behaviors start off being deliberated, requiring attention, being quite slow and variable. So you can try different options, see what works best, and being responsive to changes in the outcome. So if your context changes, if your behavior is not producing the result you wanted, you can easily change it. But once the same behavior becomes habitual, it becomes reflexive, requires minimal attention, is fast and stereotyped. You do it the same way each time, and it becomes unresponsive to changes in the environment, which can be a bad thing when you need to change the behavior in order to adapt to new environmental requirements. And one other special property of habitual behaviors is that oftentimes they're composed of multiple different steps, which are chunked together as to create a single unit of behavior. So for example, when you're brushing your teeth, you have multiple steps involved in doing that. But once you learn to do that, you kind of think of it as a single action sequence. And this chunking phenomenon is what I was really interested in studying in grad school. And I chose to focus on a region of the basal ganglia, which is called the dorsolateral striatum, which we know is extremely important for habitual behaviors. And we know this for many reasons, but partially because when this area of the brain is damaged, animals have trouble transitioning from this deliberative slow stage I was talking about, from the initial stage of habit learning to the later stage of habit learning. So what I wanted to do is figure out what's actually going on in the basal ganglia and the striatum that facilitates this learning of habitual behaviors. So in order to study this, I taught rats a basic habitual action sequence. So this would be kind of the analog of you typing the password into your computer every day, or typing in your ATM code or something like that, where you do it so many times it just happens by itself, kind of. So for the rats, what they had to learn is to press a specific sequence of 3 levers. And so for example, in this rat, they had to press the first lever, then the second lever, then the first lever again, in order to get their chocolate milk reward. And if they did any other sequence of presses, then they wouldn't get the reward. And so what I did is I taught a bunch of different rats to do a whole bunch of different sequences to see whether I see neuronal activity in the striatum that has to do with the development of a chunked habitual action sequence. And you can see that the rats did learn to do this, and not only did they learn to do it, but they also exhibited certain behaviors that made us think that they were really developing a habit. So for example, they would follow stereotyped movement trajectories. Here you can see the rat, its head is moving in the same way trial to trial when they're doing their sequence. And importantly, each rat did it differently, so they were assigned different sequences, and they also developed different ways of doing it. So they developed their own special little habit that was different from animal to animal. So what was going on with the neurons in this habit-related area of the striatum? Well, what the neurons were doing was that, so here I'll just explain this chart a little bit. Each row in this color plot is a single rat, and you can see here on the left which sequence they learned. And each of the black lines are the 3 lever presses that they did. And then the color of the color plot indicates how active the neurons were at that time. And what you can see is that across the rats, regardless of which sequence they learned and which movement they were doing, the neurons in this area were active around the start of the sequence that the rats had learned and around the end of the sequence that the rats had learned. And we think in combination with the fact that we saw the same kind of start and end activation in rats that were doing a totally different habitual behavior, rats that were running down a T-shaped maze. And again, these neurons were active at the start of the maze run and at the end of the maze run. We think that probably what these neurons are doing is responding, basically demarcating habitual behavioral units that the animals have developed. And we did a lot of things to rule out other explanations like movement. We found that this doesn't happen when the rats are doing the wrong sequence that they never learned, that they weren't rewarded for. And also what was interesting is there was a different kind of neuron, an interneuron, which is sort of a locally modulating neuron that modulates the principal neurons, that were doing the opposite pattern here. So what you can see here is that the main neurons were peaking in activity at the start and end of the sequence, and the red plot here shows what these interneurons were doing. So they were doing the opposite pattern, potentially keeping down the activity of those projection neurons while the animal was actually performing the sequence. And what was kind of fun to see was that in the cases where the rats messed up, so for example, here what I'm showing are the cases in which they started off doing the first 2 presses correctly, and after that, the 3rd press, they messed up. And what happened is that you see this activity, the start activity peak is there, and then the middle activity peak in the interneurons is there. But then when you get to the last incorrect press that they did, the end activity peak never comes, and then the interneurons are still kind of going like, oh, I guess we're not done yet. So this neuronal activity in this part of the brain was really reflecting whether the specifically this sort of abstract concept of whether the rats were performing a habitual action sequence that they had been rewarded on before. So what does this mean? You know, what can we make out of this neuronal activity? Well, we know that learning and selecting behavioral repertoires is a fundamental function of the nervous system. And we also know that over or under expression of habitual behaviors is critically detrimental to quality of life. So if you're unable to develop these habitual behaviors, and every single little thing you do in your daily life, you have to think about every step you're doing, that's going to be really difficult. Whereas if you're too habitual, you're doing these compulsive behaviors, for example, like in obsessive compulsive disorder, now this is really maladaptive because you're unable to reallocate, sort of adjust your behavior as needed in order to adapt. Similarly, in drug addiction, which is obviously the main topic of this forum, a certain trigger can cause the cascade of some habitual behaviors which lead to the drug taking, and it's a self-reinforcing cycle. So addiction is also very much another disorder in which there's overexpression of habitual behavior. Whereas, for example, in Parkinson's disease, there's a loss of learned motor action sequences that people had previously in their life that they become unable to do once they develop Parkinson's. So that's it for my little talk. Let me know if you have questions about that later. I'm gonna stop sharing my slides here. Okay, so it's okay?
Good.
We're back to the normal view, okay.
Okay, so I'm gonna try to pick up a little bit where Nune left off by showing some work that we're doing at Yale on experiments on people. And the first thing I need to do is be able to share my screen with you so you can see what I see. Okay, and that should happen shortly. Okay, hold on a moment. Okay. Sorry for the delay. I'm just going back to the, okay. So it's always good form when you're a professor to start with acknowledging all your students, which I do on my first slide. So all the people on the left were either students or postdocs whose work contributed to what I'm going to show you, and in exchange, we gave them a PhD. Okay, so this is a design of a study that we do with human subjects who are addicted smokers, and we study them with PET brain imaging. And the important things to know for the purpose of this little discussion is that our smokers come in and they're abstinent the night before, from the night before, and so presumably they're craving their cigarette. We place them on a PET scanner, which is indicated down on the left, and we inject them with a tracer, a radioactive tracer, which is necessary to perform the PET study. And at some point into the study, actually at 35 minutes into what is a 90-minute scan session, they smoke one of their own cigarettes. They give themselves the cigarette. And so for those of you who are addiction experts, this is self-administration, and we create the images over time, and we look for the dopamine response to their cigarettes. Dopamine is the neurotransmitter in the brain that is encoding reward, among other things. And we look specifically in the striatum, which is an area of the brain that the previous speaker spoke about. And if you wanted to actually observe this study, it would look something like this if you were looking into the PET suite at Yale from the control room, and you see that we're monitoring head motion. There's a picture of me when I had hair. We're filtering away the secondhand smoke so that the techs are not injured, and the person in the scanner is being scanned and about to smoke. Now, on this slide, this shows you the most important thing, of course, about doing research is the graduate student. And in this particular case, the graduate student is doing a very important job, which is not only lighting the cigarette, what you see in the middle of the scan there, the picture, but catching the ashes so that we don't burn up the subjects. Okay, here's some real data that derives from those experiments. And this is going to go by pretty quickly, so I'll show you twice. This is the same person, the brain of the same person smoking once on the top row, and then coming back another day not smoking. So just to show you what we actually derive from these experiments and the mathematical modeling that goes next, let me play the video a few times. It goes by very quickly. You should have noticed some burst of activity in the striatum, which is here on these contiguous slices in the brain of the subject, but nothing at rest when they're not smoking. So let's look at it briefly. I'm sorry. Let's look at it briefly again, just to convince you that we're actually picking something up, which we, for reasons I can't explain right now, attribute to dopamine activity. Okay. There we go. One more time, the bursts of activity in the ventral striatum of the person when they're smoking, but not when they're doing nothing. All right. Now, this is just for MIT alumni who may be asking at this point, well, what's the engineering in this? There's a lot of physics in PET, and there's a lot of radiochemistry, but for me, this is the contribution that my students and my lab and I make, which is that the models that used to be used to analyze the data that we generate were previously assuming that dopamine was at steady state. But the models that we contributed to the field are, in a sense, tuned to the frequency of brief cigarette-induced dopamine events. They're not quite as fast as I showed them, which is seconds. They happen over minutes. But since they only allotted me my 7 minutes during this podcast, I showed it to you quickly. So now here's the heart of the matter. If you look at the bottom row, and I'll show it twice, this is a male smoker. I'll come back to it. And now the female smoker is being shown on the top row. Let me replay it, because I think you'll be able to distinguish the difference that we started to see as a consistent pattern when we compared male to female smokers. I'm going to play the bottom row, look at the bottom row of contiguous slices in the brain of this male smoker, and notice that here in the right ventral striatum, there was dopamine response. Now, if we look again at the female smoker, by looking at the top row, you'll notice that a little later in her scan, she responds in the dorsal striatum. And this was something that had been, there is evidence for sex differences, but let me try to explain why this is important and to tie it back to the previous speaker who spoke about habit. So what do we know that's different about male and female smokers? Well, for one thing, men are reinforced by nicotine. And interestingly enough, it turns out that they're about 1.5 times more likely than women to stay abstinent on nicotine replacement therapy, like the nicotine patch. And this implicates the ventral striatum, whereas women smoke for other reasons, such as social reasons and the cues, and in particular focus on habit. And they turn out to be less sensitive to the nicotine level in their cigarettes than men are. This in turn implicates the dorsal striatum, which is just the type of difference that we were seeing between our male and female smokers. So there was some consistency to what we saw. And so now we're engaged in a much larger study looking at the effects of addiction, in particular still smoking, on the brain. And I think of it as a signature, a John Hancock of addiction. And we're also looking for the effect of treatments, including nicotine replacement, this is the right half of the slide, on that signature of addiction. And in particular, we're searching for sex differences in the alteration of the addiction signature caused by treatment. Okay, so I'm going to leave it there and give way to our third speaker, who is Professor Amy Meadows at the University of Kentucky. Let me stop sharing my screen so that Amy can proceed. Hold on a moment, please. Okay. Sorry, I'm having a little trouble here. Screen sharing. Okay. I think Amy should be able to proceed now.
Thank you so much. So I'm going to talk just a little bit about the clinical applications, and I'll tell you a little bit about my background. So like everyone here, I went to MIT for undergrad. I was a brain and cognitive science major. I went on to get my MD, so I'm going to talk about the clinical perspective of addictions. And specifically, I am a child and adolescent psychiatrist and a pediatrician, and so I'm going to talk about addiction in adolescence. So after I finished my fellowship, I returned home, which is to Kentucky. So I'm at the University of Kentucky. And as many people may know, I'm right in the middle of Appalachia, which is currently the heart of the opiate epidemic. If you look at heat maps for where addiction, and specifically opiate addictions, are the most prevalent in the United States, kind of right up and down the Appalachian Mountains tends to be one of the places where there is the most. So my initial interests were in early life adversity and childhood trauma and how that shaped different behaviors. And specifically when I was getting my MD at Yale, I was doing neuroimaging looking at early life adversity, child maltreatment, and the prefrontal cortex. And so a lot of what I do now also relates to the prefrontal cortex. So instead of talking about the striata, I'm going to talk a little bit about how our executive functions can modulate addictive behaviors. So we know that people do things that aren't good for them. They smoke, they don't take medications that are effective, and a lot of times people do things under stress. So there's a very complicated relationship between stress and behaviors, especially addictive behaviors. We know that adolescents have a high rate of experimentation with substances. Current statistics show about 4% of US seniors have experimented with opiates, for instance. Most commonly, they are medications that are available in their home or from their friends, like hydrocodone, oxycodone, tramadol. And medical use of opiates in kids highly correlates to subsequent non-medical use of opiates. Interestingly, the pattern has somewhat changed. In the early 2000s, more kids were using to get high, whereas a lot of what is described now is that adolescents use to relieve tension and to relieve negative affective states. But there are some good numbers that are coming out that use of opiates has decreased, although unfortunately rates of things like emergency room visits and overdoses have continued to increase. Now, the adolescent brain is pretty interesting. We've talked a little bit about the reward circuitry and dopamine being very reinforcing, but what's fortunate is that the prefrontal cortex can modulate often the effects of dopamine release, that someone knows that they have to go to work, that they are planning and have reasons to not use or not follow habitual behaviors. However, the prefrontal cortex in adolescence is undergoing a period of extreme change. So there is synaptic change, they're undergoing dendritic process pruning, neuronal membrane synthesis, and myelination, which is really important. Now this becomes important because you combine a period of experimentation with less ability to modulate the behavior, and you become what's known as all drive and no brakes. So this is a high risk period for substance use. And we know that when you use substances earlier in life, that is a higher risk for problem use as you go on. Now, I mentioned that my specific area of interest is stress, and especially early life adversity. So a concept known as adverse childhood experiences, or ACEs, which is not just child maltreatment, but a broader category of household dysfunction during childhood. And about 50% of the population attributable risk of drug abuse is relatable back to early adversity. So this is something that happens pretty frequently. And if you look at community samples, in some communities, the comorbid risk of post-traumatic stress disorder and substance use in adolescence is as high as 20%. So 1 in 5 kids have both PTSD and substance use disorders, and that's here in the United States, unfortunately. Now, what's really interesting is that if you look at animal models, that, for instance, exposure to shocks increases the reinforcing nature of opiates. And that relates to kids saying that they're using opiates to relieve tension. So similar to what rats will do. We also know that the stress state just allows less modulation of the reward pathway. So there's increased sensitivity to environmental cues rather than being able to decrease the habitual addictive responses. So I'll just briefly end on talking about my research, which I'm looking at how adverse childhood experiences affect health risk behaviors. So that includes addiction, but also perception of risk and planning, as well as impulse control, which all relates back to those executive functions. And what's been found is that there is a dose response to the amount of early life adversity that you have to your risk for a lot of these things. So to your risk for cigarette smoking, your risk of opiate use disorders. And although some of this is replication, we're also trying to use technology to ask these questions in a novel way. So there's some indications that teenagers will be more truthful when we're asking via technology, using iPads or using computers rather than asking in person, and using kind of ecological momentary assessments, so day-to-day assessments of mood and stress states and how that relates to some of these risk behaviors that they may be engaging in. And really the ultimate goal is to ensure that we're not just addressing the substance use, but also the predisposing factors that may cause somebody to be at risk for a addictive behavior. So I apologize for not having a PowerPoint, but hopefully kind of conveyed the overview of kind of what I'm interested in and what we're doing here. I'd be happy to answer questions too.
Okay, thank you very much. Let's see if we have any, if we've received any questions yet from the participants. Okay, so we do have a question for Amy, and it asks, how do you measure household dysfunction in an objective way? We The short response is that we're asking humans to report on their experiences. So really we are using validated measures of something called an adverse childhood experiences scale, where people are reporting on their own early life traumas as our marker. And that's been used in lots and lots of epidemiologic studies, as well as in smaller studies to be validated. I see. Thank you. Okay, let's see if we have any other questions from the audience. They're running to their dictionaries and encyclopedias first to formulate their questions, apparently. Well, maybe if either of you can tell us perhaps some more about anything that you learned at MIT that related to this, your choice, your professional choice, and the work that you're doing now, that might be of interest to the audience. Is that possible?
Evan, I think that there's a question for you that says, could you elaborate on some results after treatment for men and women?
Sure.
If you have them. And
yeah, thank you.
I didn't. Others. Yeah.
Okay, great. Thank you.
Mm-hmm.
So I would say that right now we've been studying in our follow-up study that was sort of indicated in my last slide for a few years, and we are preliminarily finding differences in the dopamine response. I should say sex differences in the effect of nicotine replacement on what we think is the standard male versus female response to smoking. But that's preliminary and it's not published yet, so I don't go out and bet the house, but that's what we're finding and it's consistent with our hypothesis. So what we hope is that we're finding sort of the actions in the brain that determine the difference in response to nicotine replacement that men and women see. Okay, so it looks like we have a few other questions. Let's see if we can divvy them up. So there's a question here, it's not clear, maybe it's for Amy. Are these brain and behavioral patterns independent of the nature of addiction? That is to say, smoking versus alcohol versus illicit substances. And then we're asking for, there's an ask for references, which maybe we can provide later online. I would say, for me, it's certainly the case that many addictive substances have been studied with PET imaging, and typically we understand that the dopamine response to all of these addictive substances is quite similar, because ultimately, whether it's nicotine or alcohol, the downstream effect is to funnel through the dopamine system to encode reward. And we are, with our technology, studying other addictions. Maybe one of the other panelists would like to say something as well.
I would just like to amplify that, you know, all of the substances to a greater or lesser degree are activating the reward or motivation pathway. We do know that there is a variability between how addictive certain substances are. For instance, if you smoke one cigarette, you may be at less risk of addiction than if you do one hit of heroin, but the underlying brain pathway is very similar.
Okay. So now we are finally, our participants have thrown off their inhibitions. So they've disinhibited their prefrontal cortex, perhaps, and they've sent us a slew of questions. So I'm just going to go through and we'll try to get to as many as we can. One asks, Eric Sadler asks, I was hoping to hear more about neurological changes from substance use. And I can say from an imaging standpoint, there are many drugs of abuse that are known, well, are believed to, I should say, diminish the number of dopamine receptors and other brain receptors. I think it's not entirely clear yet whether people are born that way or it's the cause, it's a result of substance abuse. Maybe that's what I can contribute. Does anybody else want to jump in on that one? Okay, maybe we'll go to the next one. So Mitko asks, what are some effective ways to snap out of addiction? That sounds like a clinical question.
Sure, I can take that. So, you know, I think that there are a lot of different models for how to treat addictions. one of the big factors is motivation. And so a lot of the evidence-based practices about addictions treatments are focused on how to kind of mobilize people's own reasons for wanting to change and their own kind of internal motivation to snap out, in the question's words, of an addiction. There are lots of effective treatments, including cognitive behavioral therapies, mindfulness-based therapies. For things like nicotine and opiates, there are agonist therapies where you have a replacement for the addictive substance. And there are also some psychopharmacological treatments of addictions that have shown promise for certain substances.
Okay. Now here's one I'm not sure, but what the answer is. Let's see what we know. So Nazir asks, what about sexual addiction? And specifically, it sounds like he or she is asking, are there differences between men and women? I don't actually know. I'm guessing that sex addiction is also dopaminergic. I don't know of any studies in my field. There are certainly studies about gambling, which is dopaminergic. Maybe one of the two of you has something else to add? Nope.
It's not my area of expertise.
Okay, yeah. All right, let's move on. Sorry. Once someone acquires a dopamine sensitivity to an addictive behavior, can this ever be reversed, or is the brain permanently changed? So I should know that from imaging studies, but I can't say that I do know the answer. I'm hoping maybe one of my colleagues is on the line and can answer. Anybody else?
My understanding is that there are certain changes that I wouldn't necessarily say are permanent, but are long-lasting. Even after the addictive behavior itself has ceased. So even after, for instance, you're abstinent from a substance of abuse, there are some neurobiological changes that remain, and that you may always be, maybe not always, but for a long time, may be vulnerable to certain cues of the substance. So seeing somebody else smoke, or when you're going through a particularly stressful period of life, you may be more likely to revert back to substance use.
Right. Okay. Thank you. I know that there are certainly, I would call it, I guess, not necessarily dopamine sensitivity, but the flip side, which is tolerance. And in some cases, in many cases, drug addictions cause diminished dopaminergic responses to drugs. And I think that that's long lasting. So here's a question from Bob, which goes back to a previous question maybe, and it's clinical again, I think, which is, how does one interrupt the addictive brain thread from cigarettes to alcohol to food? Maybe the question is more about substituting one addiction for another. All I can say is that I know from recruiting our subjects, I also study alcohol addiction. Oftentimes, these two things go hand in hand. Does anybody have another comment? Okay. And I guess this one's for me specifically. I'm being asked, would we expect gender differences, such as what I was alluding to with smoking, would be similar for adolescents? And that's a very good question. And unfortunately, in my case, I have a methodological answer, which is we're not allowed ethically to do PET imaging. That's my get out of jail free answer, because we inject radioactivity. But certainly at some point, unless these things are inborn, I should say, excuse me, then there's we would have to expect a sort of a divergence in the development of addiction. So I'm guessing, don't hold me to it, that at some point during the course of addiction, there is a divergence between men and women in their response to addictive substances. And I think I can actually say, from what I recall, that women proceed more rapidly to the habitual stage of addiction, at least with smoking. Again, my colleague who's not on the line is the cigarette smoking expert, so I apologize. This is a basic science question for Nune. Do you have any ideas or data about how to break a chunked action?
Hi. Okay. I have ideas about it. I'm not sure how much has really been. I think it's a bit harder to study in animal models, but I think in people, usually some good strategies are things like kind of forcibly making kind of basically if there's some trigger that causes you to start some chain of actions, and perhaps try to substitute a new action in the place, in the second step or in the third step, and then kind of repeat it many times over, so that it kind of interferes maybe with with the circuits that are reinforcing the existing chunked action sequence that you do have. Another option would be to just avoid the trigger in the first place. So this also, I think, works for drug addiction, is to kind of try to unlearn the behavior by kind of setting up circumstances in which you are not triggered to perform it for a long time, after which then maybe it will be easier to kind of decompose that behavior and alter it. Or so I think substitution and avoidance are both good options, but substitution with something that is good, not with another kind of behavior that you don't want to be doing, obviously, is the best strategy.
Okay, and here's a nice follow-up question from Patty Newbold, who was obviously listening and synthesizing what everyone was saying, because she asks for Nune, what role does dopamine play in chunking of habitual behaviors, if any?
Yeah, so we do think dopamine plays an important role in two ways. One is that we think that perhaps dopamine is really important for for creating the beginning and end neuronal activity that I showed you in the striatum. So some reasons we have for thinking that are that some people have found similar kind of activity in dopamine areas of the brain, which send connections to the area of the striatum that I was talking about. And also because the dopamine can cause connections between brain areas. So for example, the input from cortical areas to the striatum, which may be becoming shaped during the process of the habit learning, this can be modulated by dopamine. So, and then on top of that, what can happen is that usually the behaviors that become chunked are the ones that are reinforced. And so there's an interaction between ventral striatum and dorsal striatum, where ventral striatum is sort of participating in reinforcement learning, and then the behaviors which led up to that reinforcement may be encoded and chunked by dorsal striatum. So yes, we do think it plays a big role for sure.
Okay, thank you. Amy, I think this question is for you, from Helene Hopkins. And she says, regarding teaching replacement behaviors to the addicted, which behaviors can one teach or recommend to the addicted that can complete, maybe she means compete with opioids, to relieve the stress they are attempting to medicate or to alleviate? It's a little...
So I'd say that nothing can compete with an opioid high. The way people subjectively describe, especially the first time that they use, is that it is a transcendent experience, people who are predisposed to addiction especially. So really there is nothing. We can do medication agonist treatment that can sometimes help to hit some of the same receptors, but it doesn't have the same experience of getting high. And so really a lot of times what the treatment is learning how to manage stress and to accept negative emotions and distress rather than trying to bury it in a with substances. So oftentimes it's not that we are having competing because nothing would compare with opiates, but rather to promote acceptance.
Okay, thank you. I think I have 3 questions that I can lump or chunk, as it were, together, having to do with nicotine replacement and other means of delivering nicotine, and possibly men who do or do not respond to nicotine therapy. So I'm just going to give a generalized answer, which is to say that despite what we're finding preliminarily, I would not go so far, and I'm also not a clinician, I would not go so far as to say that nicotine replacement does not work for women. I think you have to think about these things as overlapping populations. And so we're going to learn, we hope, what the differences might be in the response, and ultimately perhaps that will lead to the development of new treatments that are better for people who don't respond to the existing ones, such as nicotine replacement. But I certainly can't say categorically that all women should throw away their nicotine patches, particularly if it's helping them. Similarly, where we personally, my group, my students and my colleagues are very interested in also studying the response to other now popular, gaining popularity methods of delivering nicotine, like vaping, but I have nothing to say about it yet. And maybe I'll leave it there. Here's a technical one, and then I'll give over the floor. Are SPECT scans as useful as PET scans to visualize how various brain structures regarding addictions? So yes, there's a very big role for SPECT. Oftentimes, SPECT does not have the same spatial resolution as PET, but there are practical advantages to SPECT, such as the use of longer-lived isotopes. So what we do in our PET center at Yale is very much in the realm of research. And SPECT, which uses longer-lived isotopes, is much more practical for circumstances in the hospital where you don't have the luxury of controlling the patient's time, but you can wait longer with the tracer at the ready. I think I'll leave it at that. And then there's a thank you to Amy, who I guess was very popular with some of our viewers. I'm not surprised. Okay, and should we take a couple more questions? Let's do this, because we're getting quite a number of them. Maybe some of them we're going to try to respond in writing, but let's see if we, okay, sounds like another clinical question. Two things. How important are genetic predispositions to addiction, and are faith-based addiction therapies such as AA more effective than programs that do not rely on calling upon one's higher power? Would you like to address either of those, genetic predisposition or treatment?
So, yeah, I can definitely start with genetic predisposition. The bottom line is that substance addictions are one of the most heritable diseases out there, as so or more than things like type 2 diabetes, which we think of as very heritable. But if you look at the variance between why one person gets it, and why another person does not. A large portion of that is due to genetic susceptibility to addiction and addictive behaviors. Which, I think just reinforces that these are brain-based disorders, even though a lot of times our culture frames addictions as something that is a lifestyle choice. These are very much related to underlying behavioral and genetic susceptibilities.
Okay, thank you very much. There are a number of questions about other neurotransmitter systems, and I don't think any of us want to leave this forum with the message that dopamine is the only important neurotransmitter, but certainly these things funnel through the dopamine system. I can say briefly and only indirectly in response to another question that we're also studying systems like the opioid receptor system, not surprisingly these days. And in my own lab, we have found some sex differences in kappa opioid receptors. And I think it remains to be seen what other sex differences we'll see in other neuroreceptor systems and in other addictions.
Yeah, and I would also add to that, for example, a big topic of research recently in the basal ganglia has been on the interaction between acetylcholine and dopamine. So on the same receptors that smoking acts upon are really critical, as it turns out, for dopamine to have its effect on the synapses in the basal ganglia, and they facilitate that effect or modulate it in different ways. So for sure, there's lots of important neurotransmitters that interact with dopamine.
Okay. So if anybody, I'm sorry, we still have questions, but I don't think we're going to get to all of them. So unless somebody is, if there's a burning last statement, perhaps I'll just wrap up. Okay? So I need to say that on behalf of the Alumni Association, I want to thank everyone for tuning into this faculty forum online. Thanks very much to the expert panelists from Harvard and Kentucky, and it was my pleasure to join them. And we didn't get to all the questions, I apologize, but we will forward the questions to the panel. And please tweet about today's chat using the hashtag MIT Better World, and send follow-up questions or feedback to alumnilearn@mit.edu. Thank you again for joining us.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.